It's 3:40. I know we're going until about 4:50. I'm trying to do math in my head here. So this talk will not take the rest of the time, but one thing that we talked about maybe is just having the residents, and I can even get off the podium and just have a little meeting if they're still, if some of them are around, and just talk about career stuff. But the goal here is to talk about translational research in a way that explains how to get from an idea all the way to commercialization and what are the decisions that you have to make. Before I dip into it, there was a question that came in online that I did not see about the dispensers on the spyglass lens. Those dispensers are not refillable. So the question was, are they refillable? How do you add 3 years on top of that? They are also not bioerodible, biodegradable, so they don't change in shape and mechanical characteristics, so that the marriage to the capsule, the position of the lens, all of that does not change over time. It's basically the drug embedded within that material that delivers over the 3 years, but doesn't change what the capsule sees, basically, on the dispensers. After 3 years, we have a different way of allowing for 3 more years of therapy that, again, we're not talking about publicly, but I'm hoping in the next year or so that we'll share some of that information. But it does not involve refilling of the dispensers. All right, let's have some fun with translational research. This is another talk where if you just want to raise your hand, or don't even have to raise your hand, just kind of shout out some of the questions. This is in many ways a painful lecture for me to give because I talk about a lot of the pain points that I went through. I show this video because a lot of times when I'm standing up and talking about our company work or device work, it sounds like it's just me free soloing up a mountain. It's all me, like it's on my back. And nothing could be further from the truth, right? There are multiple people who are much smarter than me who work on these things. It is a scary thing no matter what you're doing, if it's just one person or a bunch of people climbing, but you really need a team to climb together in order to do this the right way. So I'll talk about where does innovation come from. We talked about this again over lunch just a little bit. Steps to get started and keep it going. I'll give you some real world examples from our device lab. I'll talk about mistakes that I made. There are several. I'm not covering all the mistakes. It's just a portion of them. And then I'll close off with some tips to consider if you want to get into this space where you think of an idea and you want to take it all the way to commercialization. So where does innovation come from? I don't think this would surprise a lot of people in the room that most of the ideas, most of the things that we're using in the operating room have come from physicians. And these are physician names that a lot of us are familiar with. So Phaco came from Kelman. Drainage devices, MD or PhD. Dr. Ahmed is PhD. Everybody else that came up with a tube to date has been an MD. OCT, you have Joel Schumann here, so you know the lineage of that and how many people worked really hard to get OCT out. Lasers, Ron Kurtz, who was the power behind LensX. He now is the CEO of RxSight. He's a retina specialist trained in Michigan, and he really led the movement of femtosecond lasers treating ophthalmology. Ron Kruger, who's now the chair in Nebraska, he did a lot of the work with one of the fundamental femtosecond lasers for cataract surgery. And then MIGS devices, for the most part, have come from ophthalmologists. So Rick Hill, who has an attachment to Will. See, I think he did his training here. Is that right? Or no? I think there's some connection here. Marty Uram, who's a retina specialist, he came up with ECP. Dewan I mentioned, and then of course Barvelt with trabectome. He's a glaucoma specialist. He passed away a couple of years ago. But it's not just the fancy devices, it's also a lot of the sort of class 1 devices that we use, whether it's a Cybele chopper, Akahashi pre-chopper, iris hooks, and Fechner forceps, which I use for all of my filtration surgeries to handle the conjunctiva. He's a glaucoma specialist. And it's something I think that's really important to remember. When you think of something as you're operating, don't just dismiss it. You probably have the insights to make things better because that's usually how things get better in ophthalmology. It's usually a surgeon who comes up with it. Just keep in mind that new does not mean better, new does not mean practical, and new does not mean viable. So I get a lot of emails, a lot of phone calls, partners stopping me in the hallway and between surgeries, and it's like, hey, I have an idea. You guys know industry, they have an acronym for this, Doctor with Idea, DWI. that industry will make jokes about this. You know, the people who are in charge of business development, if they're at AAO walking around, they're like, you know, every 10 steps, a doctor will stop me and say, what about a pair of scissors that looks, you know, like that? So you really have to think through the idea and think of, from a practical standpoint, what kind of a difference does it make? Can you imagine using that hammer there? I wanted to cover a couple of concepts, and I'll point these out. There are a couple of slides here that are not intended to go too deep into, but I want to put these out there so that you can think of them if you do think of an idea. So if you have a particular problem in the operating room or in clinic and you can't think of a solution, so oftentimes you're going on a run, you're on a walk, you're sitting at home thinking, how can I do this better? There are different ways to go through it, and these are some of the common ways to go through it. You can think through science fiction, communication systems, you know, Star Trek about, you know, touching your pin and talking. A lot of the early stuff that came on with communication was based on, can we ever do that? And so using science fiction actually does work. It helps you dream a little bit. Analogies, Circuit City turning into CarMax, using a lot of the technology that they had for one business and then transferring that over to another business. First principles, this is made very popular by Elon Musk, talking about first principles. So if you think of a problem, try and break down the solutions that exist today to their fundamental and see why people made decisions to build, and don't build on top of what they built. Go to the foundation. And then exaptation, which was new to me. This is the concept that maybe you have something that works for one idea and you can transfer it over to a different idea. So feathers might be there for warmth, but they also helped with flying. So there are different ways, and again, I'm not going to go over sort of how to think through all of these. There are multiple other ways, but when I'm stuck, I often go through this list and I just think, hey, you know, is there a solution here that can help me? Is there something that exists already that can help me solve the issue? Innovation comes from identifying an unmet need. Usually, if there's going to be something that has legs, it's because there's an unmet need, right? You have to actually have a reason for spending all the time and money to come up with the solution. Is there a better way or a new way to do this? If you're running into a problem during surgery, this is how KDB came up. It was an issue we talked about over lunch that you know, it was a problem, and I needed to think of a better way to solve the problem that we were having in the operating room. Is there a safer way to achieve the goal? So a lot of times safety will drive the innovation. And the real question becomes, how do you get from idea to solution? Because oftentimes the idea is the easiest part of what you're trying to do. I found this cartoon and I thought it was perfect for this point, right? You see the beaver walking up to the Hoover Dam saying, I didn't build it myself, but it's based on an idea of mine, right? So it's really easy to have an idea of doing something, but getting it from the idea all the way through manufacturing, through regulatory, through clinical, to the final product is the real hard part. It's the heavy lifting. So how does innovation reach the patient? It all starts with an idea, this easy button idea, but unfortunately that's the easy part. The majority of inventions remain conceptual. So I'm sure it's happened in this room multiple times where you think of a solution, you talk it over with a couple of friends, and then next week you forget about it. And that's, unfortunately, that's more common than not. The major limitations include time constraints and lack of know-how. Lack of money is a big issue, and the fear of the unknown. We're not trained as physicians to go through the patenting process, to go through the clinical, regulatory. It's not something we're trained for, and for the most part, physicians are risk averse, right? We don't want to actually say, look, I'll bet 100K of my money to get this through the patenting phase, and then maybe this is going to work in the future. It's just not the way that we operate. So how can you push forward? This is another one of those slides where, again, we're not going to go over the whole thing, but I didn't have a concept for trying to figure out if an idea had legs. So is it going to be commercially viable? I'm not trained in marketing or as a commercial specialist, but Porter's 5 forces is something that's very common in the marketing business world to figure out if something actually has legs. So you look at rivalry among existing competitors, bargaining power of supplies, threat of new entrants, bargaining power of buyers, threat of substitute products. The reason I said all of those out loud is because these are words we never use in ophthalmology, right? These are just things that are foreign to us. But if you are coming up with an idea and you're wondering about, hey, does this have legs? Should I be pushing it forward? Just remember Porter's 5 forces. And then think of your idea, go into this matrix and just kind of plant it in and say, what are the barriers to entry? Do I need millions of dollars to get this through? What's the number of customers? So how many surgeons will actually be using this? How many patients will actually need this? Number of substitute products available, probably the most important question that you're going to ask early on in the process. do I want to come into the market with the 30th intraocular lens, right? It's probably not going to be a business. There's a reason why Alcon, J&J, Bausch own the majority of the intraocular lens business, and all of the others are sort of way, way behind, right? So it's something, this Porter's five force is really important to think about early on. I'm going to talk about two technologies that came out of our laboratory and use those examples to tell you what the journey looks like. The first one is a class 3 device, intraocular lens with a PMA pathway. So there are two different pathways that you should think of when it comes to regulatory. One is a PMA, which is, just think of it generically as a longer route that requires a significant investment into a phase 3 type trial. And then early stage novel material I'll talk about, and towards the end I'll talk about a class 1 device, which is more along, think of it as the 510K route. These are numbers and letters that are used by the FDA to designate something that maybe doesn't need the rigorous clinical trial of a PMA. So it's just a tier below. Clairvista is the first company. I bet nobody has heard of Clairvista. Is that the case? Anybody here independent of lunch today? Yeah. So Clairvista was one of the first companies that I started out of the University of Colorado. This was actually, went into an incubator that was funded by a venture capital group, and then I helped the incubator launch the product. Let me tell you what it is, and I'll show you an animation for this. Before that, just a couple of pictures. So standard intraocular lens on the right-hand side, Clairvista lens on the left-hand side. The blue ring is a base that is separate from the optic. And I know it's a little bit hard to see in the photo. The animation will make it look a little bit more clear, I think, to you. The idea behind this is that you put the base into the capsular bag, which is already in place here in the animation. And then there's a second implantation that happens with the optic that goes in, and that is just using a standard Sinskey hook docked into the base with a spring haptic on one side and a plate haptic on the other. And so this does a few things. It gives you stability. Right now when we do intraocular lens calculations, we use effective lens position, which is kind of a guess as to where the lens is sitting. Now with the base, what we were able to show is that that effective lens position turns into actual lens position. We know where that lens is going to sit. And you can also put this in and then do imaging and figure out exactly where it is and then change your power depending on where that's sitting in before you put the optic in. It enhances centration and rotational stability, which is something we looked at very early on. So if you're doing a toric lens, you're nailing basically the axis that you're putting it into. Consistent and complete anterior capsule overlap. It's much bigger on the outside because of the base component than a 6mm optic. And then it also has multiple barriers to PCO. So you don't have to do YAG capsulotomies as frequently, which can be a plus or minus depending on which angle you're looking at it from. The optic itself enables a few things that you can predict. So if you put in a PanOptix lens, let's say, and the patient doesn't tolerate a multifocal, now instead of having a lot of chair time and telling the patient to live with it, you can go in and do something about that where you more easily exchange it to a monofocal lens. It also enhances the primary outcome because of the ELP stuff that I talked about. And it also gives you the opportunity for future technology. Let's say 20 years from now, we figure out a way to make a lens that accommodates in a way that we're not able to today. You have an option for future technology that hasn't been contemplated to put it into this space. So there are a lot of pluses to this. The modular design allows you to think of intraocular lenses in a different way. The second technology that I want to talk about is a material technology. So very different value proposition than coming out with a device and launching it all the way through commercialization. In this case, we were thinking of an unmet need, which is the hydrophobic acrylics that we mostly use in ophthalmology. There's usually a balance between the mechanical stability of that lens, how stiff the haptics can be with the capsule, and then injectability. Can you still put it through a 2.4, 2.2mm incision when you're doing clear cornea phaco? That balance usually leads to losing one versus the other. Sometimes the lens isn't as stable, or sometimes you need a much bigger incision than what you had initially desired. So potential solutions, I started looking at different materials with colleagues at the University of Colorado, and there was a group of materials called shape memory polymers, which had not been extensively explored in ophthalmology. So we partnered, we started a small company called Shape Tech that spun out of the university, and that's the second category that I'll cover. So setting up for success, breaking through from conceptual to development requires a time commitment that often starts with research. So this is the boring part of innovation. This usually is me sitting in my loft with my laptop in front of me, and I'm looking at I mentioned Duke Elder at lunch. So almost, I would say 95% of the things that I thought were novel, I would open up Duke Elder or look at AJO from like 1970, and that idea was there. Right? Most of the things that you think are novel have probably been tried, tested, whatever it might be, and they're not as novel as you think. You have to do that research beforehand. You also have to try as an ophthalmologist to define the unmet need. It sounds like it's like not a very interesting thing to do, and in fact, in my opinion, it's not. But you have to do the market research to say if you have an idea, is it going to be something that venture capital, other investors want to put money in? And then eventually, if it does make it into the market, is it something that you would use? And that's where we have the advantage, right? So when we're inventing and we know what surgery is like, we know what the eye requires, we can usually answer that question if we're being honest with ourselves. Is this something that you would use if it was available to you today? Has it been attempted before? This prior art search, that's what it's called. So if there's something that exists in the past, it's prior art to what you're trying to design. I've made these mistakes where I've gone as far as filing for a provisional application and then abandoning it because I found something somewhere that spoke to it. Can you build it? Is it also something that's so pie in the sky that you can't do it, or is it manufacturable? Can you actually go through and talk to people who manufacture things and see if it's possible? And if all is okay, it's time to protect your idea. So it's time to start talking about patents. I always like to throw some of these fun patents up here, because when you start doing prior art searches, when you start looking at patents, especially in ophthalmology, there's some really strange things like glasses for chickens, which is a real thing that was filed and granted. The middle one is a hat that tips by itself. So if you see somebody, instead of greeting them, you really like that idea. I like that. And then the last one I think is actually pretty practical. I would use this on a Southwest flight where you just put a hammock and you fall asleep in the hammock. There are a lot of strange ideas when it comes to patents, and if it's novel, it's going to get protected. There's nothing about saying that it's practical, right? So if it's something that's unique and novel, you can probably get it protected. It's important to start out on firm ground, so you don't want to start an idea and be 5 years down the road and not have something that's protectable, right? You have to make sure that what you have is firm, and you should have an expert opinion about that. Seek a patent attorney who's familiar with the space, because you're going to save a bunch of time. You don't want to take 2 months out of your life to educate somebody up to the point where they understand ophthalmology. We're fortunate that there are a lot of ophthalmic experts in patent offices because there's just a lot of activity in patenting ophthalmic ideas. Talk to others who've been through the process. Talk to a lot of the names that I mentioned earlier in this talk. Almost everybody that I've approached, even much earlier in my career, if I just called them or emailed them, I can't remember somebody ever saying no to talking to me, right? Just, hey, how did you do this? How did you think of this idea? How did you come up with this application for a patent? People are usually pretty willing to speak. Try not to be too narrow in your focus from a patenting standpoint, because you want to protect as much of it as possible. But if you're too broad, then you really aren't protecting anything, right? So you have to find that sweet spot. And then freedom to operate, that's what FTO stands for. You need to make sure that you're not just protecting something novel, but once you have a patent and it's protected, with that novelty is protected, you have to make sure that there isn't anything else out there that can block your ultimate idea, right? So it's an idea of, yes, it's novel, but I can't make it or I can't commercialize it because somebody else has something that protects a component of it, which would still block me, right? So the FTO part is an extremely important part, and that's where the lawyers come into it. You have to do the heavy lifting. Even if you have a patent attorney, this happens every time we file for a provisional application, the first stage of patenting, we're heavily involved. We're doing a lot of the writing. We know a lot of the literature. We know the lingo around it. So we're doing a significant amount of the heavy lifting when it comes to the application. And then money is often the major limitation. But if you have something that's really good and you think it has a lot of potential, filing for a provisional application, if you do the heavy lifting, can be $3,000. So, I mean, it sounds like maybe a lot for the residents at this stage, but it's not so much money that you're locked out of the process. So device build. Now that you've protected your idea, you have to start thinking about, how do I build my idea? We hear a lot about the tech space where, Apple started in a garage. Multiple companies that are in the internet business started in a garage. I still don't know of an IOL company that started in a garage, so we live in a different space. But we do have some limitations from a contract manufacturing standpoint that I think are really important. You should, as much as possible, if you're in practice and you're thinking about making something, try and find somebody that's local so that you can drive and meet them face-to-face, or they can come to your office and meet face-to-face. That interaction, that proximity is extremely important, so you can kind of keep your finger on the pulse of things. POC, proof of concept devices, can be very simple, so you don't have to make the final product. If you're thinking of something where you want to get investment, you're thinking of a way to get people interested, you can come out with a simplified form. It doesn't have to be the final form, and it's something you can do with most medical device manufacturers, and you have dozens. I looked it up actually before I came here. There are many, many in Philadelphia that can do it for you. We're fortunate in Denver to have the same ecosystem. Seek larger partners. This is one point where I made a big mistake early on. If you're teaming up with somebody and you're going down the road with them, make sure they can follow along to the subsequent stages of innovation, that they can take you from 10 devices in a day to 1,000 devices manufactured in a day, so that they can scale up production so that you can launch something. That can be a limitation down the road if you start having to think about transferring plans and know-how and all of that to a different manufacturer. It's better to start early on with the bigger manufacturers when it makes sense. So what are the different sources of funding? I get asked this question quite a bit. There are a lot of different sources of funding. One is you just self-fund, right? The pros on it is that you don't have any dilution. Nobody's taking a share of it. It's really not a headache, except that, you know, you might be dealing with your significant other to say why you're spending money on a new idea. On the con side, easier said than done, and you almost never know when to stop when it's your own money and you're passionate about it. That's why you don't see a lot of very wealthy individuals like Elon Musk, for example, putting all of the money in when he can do that. He usually goes after investors to join with them because it's good to have that counterbalance. You can go to friends or family, more money if you're pooling money, but just don't ever do business with family. So I'm just going to stop there. That's a big con. Angel investors, deeper pockets, and sometimes good advice. Sometimes you can find people who know what they're talking about and they're angel investors, and you can have sort of a partner there, which is really good. On the con side, now you're getting dilution. So that angel investor isn't giving you money for free. They're expecting something, some portion of the idea in return. Sometimes you get bad advice. Sometimes the angel investors are just good with the money and they don't really have good advice from a company standpoint. And sometimes they have unrealistic expectations. If it's an angel investor who's not a traditional angel investor and has done a lot of angel investing, they might think they're getting a return on their investment in 6 months or 12 months. And that just creates a lot of havoc because it's with medical devices, it can be 5-8 years before they see anything. You can go after university funding, no dilution, higher percent of success in getting that money. But it's typically something that you're doing on your own even after you get the money. You don't have an ecosystem around you. Universities are not set up for companies and for getting technology done. Some of you might be familiar with SBIR. This is a small business grant that the government gives out. A high rate of success, but it's cumbersome, time-consuming. It's a lot of paperwork. And then the traditional one is venture capital. Serious partners, usually very knowledgeable. It gives you a great deal of validation when a venture capitalist who, if the company or the group is a known investor, it gives you great validation that if they think you're real, then you might be real, right? On the con side, welcome to significant dilution. So the way that it usually works is with each round of funding that you bring in, you're losing about 40% of your company. Right? So you start off, lose 40%, next time you're losing 40%, next time you're losing 40%, right? So you just keep doing that. And when you need millions of dollars to get an idea across the finish line, it's usually the only way to do it, right? But it is a significant con. So let's go back to ClearVista. With ClearVista, there had never been a modular intraocular lens on the market. Or developed. To our knowledge, and that's still the case today, there's been no lens that looked like this with a base that had an optic that there are some that are accommodating right now, like LensGen and a couple of others that are out there, but nothing like this where it was designed for primary exchange and for stability. So we actually had to come up with a lot of things along the way that nobody else had done before us. So we went after venture funding after doing a roadshow. We went through 104 different designs, 8 device builds to come up with something that we felt very comfortable with. That was a year of work in itself. We also had, these are things I didn't think of early on in the process. We had to come up with our own material because Alcon wasn't about to give us Acrosoft or Clarion material to do our thing. We had to actually make our own material. and that was a heavy lift. We also had to come up with bench stability testing to show that what we were saying about ELP was true. So we came up with our own bench test. We published this in papers to show that we're doing the science that is needed. We went through animal model testing, injector optimization, because there was no injector specifically for the base. We visited with a bunch of key opinion leaders. We went through regulatory, and then we planned for proof of concept in human studies. All of this was done before setting one foot into the operating room. So it took a very long time, a year and a half basically, to get to that point where we started doing our initial implantations. With ShapeTech, a little bit different because that wasn't going to go into patients right away. So we were able to use university funding and grants from the university to iterate hundreds of formulations to come up with optical quality, stability, injectability, everything we were looking for from the material. By the way, these two companies were not related, so we couldn't use the ShapeTech material for ClearVista. We had to come up with our own material for ClearVista. Injection testing and injector innovation was done at the same time. Manufacturing and scale-up was done in-house to show that it was feasible. We went through animal studies, and we were prepping for FDA studies. From a regulatory standpoint, we get almost no training as ophthalmologists about what the FDA does and how to do things. A major factor in what we do in the US is dictated by the FDA, so it's a really important step. It's difficult to learn each system globally because not everybody is just like the FDA, everybody's a little bit different. The FDA website is a good resource. There's a lot of information, so if you're just starting out and you're motivated, you can learn a lot from just going to the website. Talk to industry colleagues. Most industry members are very happy to talk about regulatory, and they have their own regulatory experts. And then seek an FDA consultant. So I would have done things differently with my earlier ideas where I brought in a regulatory expert from day one because they can shape everything you're doing, from how you're going to fundraise, how much you're going to build, how far you have to go with animal studies before you go into humans. All of this is extremely important. So FDA consultant, day one, start thinking about talking to them. Through all of this, you have to focus, right? There's a lot going on. If you're like me, you're in clinic, you're in surgery, have a family, trying to do these ideas, and focusing is extremely hard. So you can fall for squirrels, start thinking about new ideas, not chasing down the idea that you have today. And all of this is happening when we're starting to think about doing a clinical trial for ClearVista. And you can't do this in the United States, right? It's something where you have to start going ex-US to do some of the initial low barrier regulatory areas where you can start doing implantations. And so I was on a plane a lot. I was visiting multiple countries. Glenn Sussman, who you saw his picture earlier with Spyglass, he was also with ClearVista. He flew 1,000,000 miles during ClearVista time. Separate ORs, separate KOLs, visiting all of these centers and trying to get them to do the implants. And once they were doing implants, visiting each time they were doing an implant. And you can see for ClearVista, we operated from between Auckland and Tijuana. We were all over the place. For ShapeTech, we completed our proof of concept studies. We did in-lab. ISO standards are basically the roadmap to how you're supposed to do things from a build standpoint. All of this was Colorado-based. We didn't do a lot of travel, so it was a much different story compared to ClearVista. Spin-out was completed to an off-campus facility along the way, so we got off-campus, but we were really on campus. It was just one street away. It was a facility that was owned by the university. We started meeting with strategics, contract manufacturers, meeting with KOLs, and exploration of the real potential. So it was a much different story than what we did for ClearVista. And then the big question is, when do you partner? Who do you partner with? How do you go through that decision-making? You could go to strategic companies. You see a bunch of them listed on the right-hand side. It's great affirmation for your idea. It's like getting venture capital money. If Alcon says they're interested in something, then it must be real, and you start getting excited about the potential. But strategic companies are very slow to execute. Once they have an idea in-house, they don't operate as fast as you do. You also completely lose control in most circumstances where the company that you were partnering with is running the show rather than you. And I only recommend this for ideas that are very mature. Right? So in the case of KDB, for example, I partnered with New World Medical towards the end of that pathway when we were done kind of thinking of how the manufacturing process is, and they still had a significant amount of input into that and made it better, but I tried to wait as long as possible. Venture or other investors can be a great partner. It's exactly what I listed above. with the potential to remain engaged and build value. And that's what we're doing with Spyglass today. You could also go it alone, but it's much harder to go it alone without partners for the long term. You eventually need somebody on board. So the decision for ClearVista after we got to the initial first-in-human data was to sell the company. Tough position to be in because we're thinking, do we go all the way through commercial approval or do we offload it to a company that can do it faster than we can? The cost of phase 3 was going to be in the tens of millions of dollars, and that was one of the drivers. We were also only 5 years into the company, so we were able to exit the company after 5 years, which is pretty good from a startup standpoint, and we were excited about that part. Alcon has the biggest commercial potential for IOLs, so we thought from a royalty standpoint it also made sense for the university. And they had a family of next generation lenses that we thought would team up well with our base. The price was right. Our venture partners were very happy with it. It felt right at the time. We had other projects that were going on that we wanted to spend more time on. Now, has anybody seen ClearVista Medical commercially? So we sold it in 2017, so it's been 7 years. And what ended up happening is Alcon started the work for phase 3, and then PanOptix came out. And PanOptix changed their world, right? It commercially became really important to everything that they were doing. So the value proposition for those that were internal changed. And also the stakeholders internally that were champions for ClearVista, as what happens usually in industry, they moved on to other jobs. So the internal advocates were less. So what they're doing right now, it's basically a patent play. They own all the patents around it, still paying royalties to the university, right? But not something that I think you'll see commercial. Which is sad, but I understand it. ShapeTech, we actually ended up partnering with J&J because they had all the manufacturing know-how and we could learn from them. That was probably one of the more important decisions that I made in my roadmap to where I am today. I was able to work with the team face-to-face on a regular basis to learn how they thought about developing materials. It was good for them because they got to work with a novel material, and that influenced a lot of what you're seeing today from J&J. So huge success for us, huge success for them. Now let's talk a little bit about a third option. I'm going to take a look at the time here and make sure we're okay on time. So I talked about the first two ideas, and I wanted to walk through KDB a little bit more just so you get an idea of how we carried a Class 1 device through all of those steps. And I'll try and go over this a little bit more quickly. So the idea for KDB was developed over a decade ago, and we did much of the development in-house. I was able to work with a contract manufacturer who made several iterations, and then through that process, we decided to start working with New World Medical, who had a huge footprint in glaucoma. And I'll talk a little bit about excisional goniotomy. The history of goniotomy is very long. It's been around for over 100 years in different forms. It was thought initially to not be good for adults, but good for kids. That turns out to be a fallacy. That was a dogma that existed for a long time, and I think we've proven that that's not the case. But the idea for KDB had nothing to do with surgery. So I do a lot of imaging research, and we had an imaging device that I was really curious to use for trabecular meshwork imaging, and we didn't have a good way to collect trabecular meshwork. So we couldn't collect a full strip of trabecular meshwork. What I ended up doing is making a device that allowed me to peel the trabecular meshwork by using parallel blades. So I was creating parallel incisions just like KDB does, but I had to iterate several times and figure out what the sweet spot was. And what we found was that the sweet spot to work on was the ramp. What is the angle of the ramp? We needed to lift the trabecular meshwork so that it was above its natural state, and that took the wet tissue paper-like trabecular meshwork and placed it on stretch so that the incisions, the parallel incisions, worked. Does that make sense? So if you try to cut wet tissue paper and it's not on stretch, it just kind of crumbles up and it kind of folds. That was the big sort of aha moment, was to create the ramp to lift up. And that ramp required multiple iterations from an angle standpoint, and also the width of where the blades were sitting. If you're a little bit off, and in this space, you don't have a lot of space, really. You're talking about 350 microns or so. Once we got to that sweet spot and we made it, we got really excited because we were getting a lot of strips of trabecular meshwork for doing our imaging studies. And it was really deep into that where I kind of thought, I was just sitting down thinking like, this would be great for surgery. Maybe I can use this for surgery. So that was the genesis of KDB. We went through even more iterations after working with New World Medical. We perfected the manufacturing process, which was a very big lift to get the blades to be sharp and to cut as we wanted it. And then we started doing some histology studies to compare it versus like an MVR blade to see what's the difference histologically. And this paper was published in 2013, so it gives you an idea of how long this work was going on. We were able to show that KDB definitely excises trabecular meshwork. It does it better than some of the other devices. This study also included trabectome, which is a cautery device, and we showed that we don't get the collateral damage that you get with trabectome. And that led to commercializing the device late in 2015. So this is, some of you might know John Berdahl. This is him doing his first case. And he's doing the outside-in technique, going from left to right, and then reversing course and going from right to left. And trabecular meshwork is very sticky, right? So at the end of this case, you'll see the trabecular meshwork is stuck right around the iris root. Again, this was his first case, so he wasn't as adept at getting it to wrap around the foot plate. It just goes in with MST forceps, and you can see the strip of trabecular meshwork. And then you see the reflux of blood on the left-hand side there. This is Paul Singh doing his first case. And you can see he does a little bit of a longer pass right to left, and then reverses course left to right. And you can see in this case, the trabecular meshwork is on the foot plate. So it actually comes out right there, comes out with the device as he's coming out. So I was pretty excited when we first started handing these. Most of the first cases were done by friends, so I'd send them the device and say, hey, take a look, videotape it for me, let's try it out. And got pretty excited after the launch in 2015, where we started seeing a lot of adoption. And that led to a lot of studies. I'm not going to go over most of these studies. I just want to show that one of the most important things right now in glaucoma and angle surgery specifically is that we need a lot more data. It's becoming an issue with reimbursement, and one of the big strengths of KDB is that we have a ton of papers. We have over 100 published papers, and it's things varying from standalone for 6 months, prospective studies for 12 months. We've also looked at our own results from the University of Colorado showing that most patients, regardless of what their starting pressure is, they can still get down to around 14 millimeters of mercury. We've also done this in angle closure in multiple sites to show that really for me, excisional goniotomy is standard of care for angle closure. All of the patients get a dramatic decrease in their pressure and they do really well. My partner, Mina Pancheva, does a lot of uveitic glaucoma, and she looked at her results, and you can see here pretty great IOP lowering for uveitic glaucoma. The question came up during lunch about GAT versus KDB. GAT is typically thought of as a really good treatment for uveitic glaucoma. That's one of the reasons we did this, and we showed that you get equivalent IOP lowering if you're doing GAT versus if you're doing KDB. You still see essentially the pressure coming down to the same level. This next study here, see if I can come up. It's always a little bit slow to come up. This was a really important point that we needed to make with KDB. There aren't a lot of randomized controlled studies in the MIG space. A lot of times you do the FDA approval versus phaco or versus whatever is required for that specific class, but you don't see a lot of head-to-head trials in MIG. So we started prospective RCT comparing KDB phaco versus iStent phaco. And this became extremely important when the reimbursement issues came up last year. So there were some of the Medicare carriers that were questioning the efficacy, and we were able to share the RCT data with them, and they changed course, right? So there was a question about coverage, and now it's full coverage. They just came out with another LCD, which is the parameter around reimbursement, last week, completely fine for canaloplasty and for goniotomy. So a lot of the data that we're getting for these devices is really helpful. In this case, KDB won from an IOP lowering standpoint numerically, but also there's a significant, statistically significant reduction in medications in favor of KDB versus iStent. So we still have a lot to learn. Trabecular meshwork analysis is something that we don't do very well. So we went from the lab to the clinic. That's how the idea of KDB came, and now we're taking it back to the lab. So what we're starting to do is collecting trabecular meshwork in the operating room. I'm getting samples sent to me from around the country. This sample was sent to me by Ron Feldman and Devinder Grover. This is a post-GAT trabecular meshwork that reapproximated, that basically closed And then they went and did a KDB, and they sent me that trabecular meshwork, and you can see some of the scar tissue. That's what you see on the bottom there. So it's not typical trabecular meshwork, but it can still heal over time. There's some desmetization there as well. This is a patient that I'm doing here, had an eye stent, and I noticed in clinic that the eye stent was displaced backward. So I went in and took out the eye stent and decided to do a KDB. As I was doing the KDB, it felt different to me. It felt like there was some resistance that I typically don't feel when I do goniotomy. And you can see here, you can see that trabecular meshwork strip right in the middle, but you also see a film, a piece of tissue that I'm gonna focus on here in a little bit that was vascular. And this was the first time that I noticed this with eye stent. And then we started noticing it with all implants in the angle. So this isn't something that's unique to eye stent. What's happening is basically what happens when you put an Ahmed or a Barvelt underneath tenons. There's always a capsule that forms. It turns out the same thing happens in trabecular meshwork. We published this in Journal of Glaucoma. Anytime that I go in and I do a post-eye stent eye, we collect the tissue. We sent a series of these in for the paper, and we see exactly the same thing. There's desmetization, there's vascularity, and there's an envelopment of the device that happens over time. So we need to get smarter on this and see why this is happening in the angle so that we can stop it. The same thing can happen post-KDB. So you go in and you remove the trabecular meshwork. You see, in some cases, desmetization. It happens less when there isn't a foreign body, when there isn't a device in there, but it can still happen even without a device. This is what the TM looks like on the right, and you can see on the left-hand side that fibrotic tissue that shouldn't be there in the trabecular meshwork. So the previous dogma espoused by generations, by decades of ophthalmologists is that you can't do goniotomy in adults, and I think that has been wholly disproven. There's a lot of that in ophthalmology that I think with data we can disprove. And I won't go over this for sake of time, but a lot of stones are cast when you come out with a new idea. These are some of the historical things that made me laugh when I was kind of reading how people went through hardships. Musician unions delayed the playing of recorded music on the radio. I don't know if you guys heard this, but they didn't feel that that was right. The Horse Association of America for many years fought against the tractor. So there's, whenever there's something that's been ingrained for a long period of time, there's usually a lot of resistance to moving it forward. All right, I'm going to finish off with a couple of things here. So the innovation triad, clinicians, basic scientists, and business leaders are needed for any idea. You can't just do it alone as an ophthalmologist. So for almost everything that we do, like I said, it's not a free solo, a lot of team members. On the left-hand side, this is the Clarivista team. A lot of them are working with Spyglass, so a lot of relationships are pretty solid and they're maintained. That's my chairman on the right-hand side. So that's me on the cover of a magazine walking with him. I always like to say that he's very happy because he's getting royalties based on these devices, so he hates it when I show that picture. Does anybody know who this is? It's Raphael Chan. He's in charge. He was in charge up until recently of the IOLs for Alcon. So a big part of their business. He's now in charge of their phaco machines. But for a long time, he was with New World Medical, and he helped with launching KDB and other devices, and very solid friendship. I'm hiking from Crested Butte to Aspen with him next weekend. So there's a lot of good relationships that happen in these industries. And the picture right above that, that's the New World Medical team. That's the COO of New World, Khalid Mansour. So a lot of innovation happening at the university. What I wanted to show you is sort of, okay, how do you think of an idea? What are some of the pitfalls? We've been through a lot of different things, a lot of different companies. I have colleagues who form companies like 2C Tech. We have hundreds of patents that are filed, 100 granted patents, 12 startups to date from the university, 4 innovators of the year in our department, which is great for a Department of Ophthalmology on a huge campus of other specialties. Over $200 million in outside investment with 4 commercialized devices. We've treated hundreds of thousands of patients with our devices, and we feel like we're just getting started. I've had the great fortune of working on several devices with different companies. KDB, I think most of you have heard of. Spyglass, we talked about. I had great fortune of helping with Streamline, which has been a really fun thing to help New World with. And of course, Clarivista Medical, and there are other things that are in the pipeline that I'm certainly excited about. So 5 tips: navigate your reality, academic versus private. Think about some of the pitfalls. Patent and non-patent prior art search. Always make sure you're on firm ground. Consider regulatory early in the process, as I said. It's really important to get the right type of advice. Build the right team. This is a long road, 6 to 8 years. You want to make sure it's people that you don't mind have dinner with, right? You need to make sure that you can hang out with people and still enjoy life. And then remember the innovation triad. You're not going to do it alone. You certainly need a team around you. Don't forget to have fun along the way. And I'll stop there. Thank you. So I think from a timing standpoint,